Hello students, this is Mrs. Shweta Laddha from Fergusson College, Pune. If you remember, we had started with our chapter 3, Inheritance and Variation. Last time we had discussed about Mendel, what type of work he did, how he carried out his work. We also discussed about certain things that are used, certain definitions that are used while studying this chapter. And we had started with monohybrid cross. In monohybrid cross, we saw that how two parents, pure parents, with one pair of contrasting characters are crossed with each other, and the produced offsprings, they show a phenotypic ratio of 3:1 and genotypic ratio of 1:2:1. Today I am going to revise what conclusions he drew from monohybrid cross. After that we will study about dihybrid cross, back cross, test cross, and so let us start with our PowerPoint presentation. Yes, students. So one more time, I would like to revise the conclusions of monohybrid cross. Mendel concluded is the unit of heredity. He did not know what is going from the parents to the offspring, so he called that term as factor, which is passed from the parents to offspring. Characters like height, seed, shape, color, they are inherited separately. He saw that they are not mixed because he saw that in F1 generation, he had obtained capital T, small t. Capital T was for tallness, small t was for dwarfness. They had come together. F1 generations though were tall, but when F2 generations were formed, when it was allowed to undergo selfing, they separated from one another. Capital T separated from small t, and in the F2 generation, you could see that dwarfness reappeared. So he wanted to say that they are inherited separately. They are not mixed with each other. Each factors, they have two alternative forms, tall, dwarf, round, wrinkled, which they are called as alleles. Also they may be dominant, recessive, and so on. One trait, it is dominant. For example, in this example again, tallness is dominant, dwarfness is recessive. In an organism, inheritance of each character is controlled by a pair of factors. Any character, suppose I am saying tallness, it will be controlled by capital T, pair of allelic pair, or capital T, small t, allelic pair. If I am saying, say, another will take round will be represented by capital R, or capital R, small r. If I say wrinkle, it will be represented by small r. Thus, in an organism, inheritance of each character is controlled by a pair of factors. And this pair of factor, one is coming from the father and one is coming from the mother. So one comes from the female parent and other comes from the male parent. He also concluded that F1 hybrid factors do not mix with each other. I told you, F1 hybrids, monohybrids, capital T, small t, they are not mixing with each other. It never happens that this small t is staying with capital T and say in next F2 generation I will become little bit tall, or the tall will say I will become little bit dwarf. No, they do not mix with each other, and so dwarfness is again appearing in the F2 generation. The factors, they segregate while forming gametes. Again and again I'll explain you this concept. Don't worry, even in dihybrid cross I'll explain you this concept. The organism that receives the recessive factors from both the parents expresses the recessive trait. Yeah, because recessive factor can be expressed only if it is in homozygous condition. So only the plant which is receiving small t in homozygous condition from both the parents, it is obtaining the recessive factor, then only it will form a dwarf plant. Now, let us see the first law of Mendelism, which is called as law of dominance. It is the first law of inheritance. Now students, whenever laws or definitions are asked in the exam, you should not change their language. You should write the laws or definition as it is, word to word. Of course, you don't just learn it by heart. You try to understand the meaning, but you cannot change the sentences. Let us see. So in a cross between two organisms, pure for any pair or pairs of contrasting characters, for example, in our case, capital T, tallness, tall plant, small t, another parent, dwarf plant. So it is a cross between two organisms, a tall organism and a dwarf organism. The character that appears in the F1 generation, in F1 generation, which character is appearing when it is F1 hybrid? It is a monohybrid. Capital T, small t is the genotype. What is the phenotype? This plant is tall. So which character is appearing in F1 generation? Tallness is appearing in F1 generation. It is dominant. And which is suppressed? Dwarfness is suppressed. So that is recessive. Example of dominant traits: black coat of guinea pigs over white, the black skin coat color of rats. Guinea pigs, here we are talking about rats. It is a dominant character over white coat. Curly hairs in us, it is dominant over straight hairs. Brown eyes are dominant over blue-green eyes. Pearly jawar, I think we call it as the normal pearl color jawar. It is dominant over the chalky jawar. Chalky jawar, I think we call them as the hybrid one, which is whitish in color. So that is the recessive one. However, there are many cases where dominance is not complete or it is absent. Up till now, what did we see? We saw that when two pairs of contrasting characters, two parents with pairs of contrasting characters are crossed with each other, one character is expressing itself in F1 generation, that character is a dominant character, and the character that suppresses itself in the F1 generation and will express only in F2 generation, if it is in homozygous condition, then we call it as a recessive character. But this law of dominance, it is not true everywhere. There are many cases where dominance is not complete or it is absent. We will study about this in incomplete dominance and co-dominance, which is a part of post-Mendelism afterwards. Now let us see what is the importance of dominance. Why it is so useful. Any harmful recessive character, if it is a recessive and a harmful character, it can be masked by the dominant character. For example, color blindness. Color blindness is caused due to a recessive gene which is present on the X chromosome, and it will be masked because of a dominant gene. Even hemophilia, even diabetes, and a form of idiocy. So these diseases, these are caused due to recessive genes, and if a person is having a dominant gene of the same, that person will be a carrier, but he or she will not be a sufferer. Basically, it is possible in females. How? We will also study this in detail later. Law of dominance is significant, but it is not universally applicable. It is true, it is significant, but not truly or universally applicable, because I told you in co-dominance and incomplete dominance, law of dominance does not work. Now let us see what is a back cross and a test cross. It's very simple, students. A cross of F1 hybrid with one of its parent. Now let me show. Suppose our parent was capital T, tall parent. But this was a parent one. It was tall. The genotype was capital T, and the phenotype is tall. And the other parent is small t, and the phenotype is dwarf. Correct? In F1 generation, what we are getting? Capital T, small t. Of course, the phenotype is tall, but the genotype is capital T, small t, because it is a hybrid. Now, if the F1 hybrid means this is crossed with any of the parent, either this parent or this parent, then it is a back cross. Going back. Having a cross with its parent is a back cross. But listen carefully. If F1 hybrid is crossed with homozygous recessive parent, I'll show with some other color. So if this F1 hybrid is crossed with the recessive homozygous parent, then only we call it as a test cross. So if a cross is of a hybrid with tall parent, back cross. F1 hybrid with dwarf parent, it is also back cross. But if a cross is of a F1 hybrid with homozygous recessive parent, then we will call it as a test cross. Let us see. After this, we'll discuss about law of segregation, which is second law of law. It is also called as law of purity of gametes. About test cross, I'll give you an example after a minute, after some time. Let us understand this law first. So members in an allelic pair in a hybrid. What was our hybrid? Capital T, small t. I'm giving you the same example again and again so that it is easy for you to remember. Members in an allelic pair in a hybrid remain together. Capital T, small t remain together without mixing with each other. Do not mix with each other. Tallness will not say, thoda sa maza tallness tu ge. Dwarfness will not say, give me your dwarfness. No. They just stay together. They do not mix with each other, and whenever gametes are formed, they separate or segregate from each other. So whenever gametes are formed, capital T will be segregating from small t. This is called as law of purity of gametes. I hope you have understood. Members in an allelic pair in a hybrid, capital T, small t, remain together. They remain together without mixing with each other, and they will separate or segregate during gamete formation. Does gametes receive only one of the two factors? Yes, here gamete is receiving only capital T. your gamete is receiving only small t, and they are pure for that trait. Yes, this is pure for tallness, this is pure for dwarfness. Therefore, it is called as law of purity of gametes. It is the second law of Mendelism. First was law of dominance. Second law, we are saying that members in an allelic pair stay together without mixing with each other, and they segregate from each other during gamete formation. So, it is also called as law of purity of gametes. Can be asked in the exam for 2 marks. All sexually reproducing organisms are in diploid and the gametes are haploid. Of course, we know this. This law is universally applicable. First law, I told you it is not universally applicable, but second law is universally applicable. Now we are coming to the example of test cross or back cross. Why is a test cross conducted? Test cross is conducted to know the genotype of or a plant or an organism when we do not know. Suppose we have this purple flower and we do not know its genotype. So it may be possible that phenotype we can see it is a purple flower, but we do not know what is the genotype. So it can be capital R, or it can be capital R, small r. It can be both. So when we are crossing this F1 hybrid plant, this is say F1 hybrid with homozygous recessive parent and homozygous. Here we do not know the dominant phenotype, genotype is known. Now if we are crossing this with a parent which is capital R, that is dominant, and we are crossing it with a recessive parent. If all the flowers in the next generation are produced violet, we will say that this plant, it is having homozygous condition. But if the flowers are produced, 50% you see here, capital R with small r gives violet and capital R with small r is giving violet, 50% are violet and small r white, 50% are wire. So half of the flowers are violet and half of the flowers are white. Such condition, unknown flower, what do we conclude? It is a heterozygous condition. Heterozygous condition, it will always be 50-50 ratio, or we can say 1:1 ratio. It is a test cross. Test cross, why? Because it is a cross between F1 hybrid and it is a cross between the homozygous recessive parent. And what is the ratio obtained? Ratio obtained is 1:1. But if suppose the F1 hybrid is homozygous, and if we are crossing it with the parent again which is homozygous, all the plants produced will be capital R, that is violet. So unknown flower in this condition is homozygous dominant. So what is our interpretation? Interpretation here, if 50-50, 1:1 ratio is obtained, our interpretation is that the genotype of this F1 hybrid is heterozygous. If all of them are violet, then our interpretation is that the F1 hybrid, it is homozygous in condition. So students, again I repeat, cross between F1 hybrid and dominant parent, okay, dominant homozygous parent is a back cross. Cross between F1 hybrid and recessive homozygous parent is a back cross also and a test cross also, because the ratio obtained there is 1:1. Why is such test cross done? Mendel devised this cross to verify the genotype of F1, F2 or F3 generation. Just now we saw the example ki we did not know the genotype of that flower. Phenotype was visible, violet color. But to find out the genotype, test cross is done. Because if hybrid is tall, it is not sure if the hybrid is a homozygous tall or a heterozygous tall. We studied the example of violet flower. So test crosses are done. Always remember student, the ratio obtained in monohybrid test cross is 1:1. Ratio obtained in dihybrid cross will be 1:1:1:1, and ratio obtained in a trihybrid test cross, it will be 1:1:1:1:1:1:1:1. So how many 1? 1, 2, 3, 4, 5, 6, 7, 8. 8 1:1:1. This is why test cross is done to identify the genotype of F1 or F2 or F3 hybrids. Now let us start with a dihybrid cross, and after that we'll also discuss about Mendel's third law of independent assortment. Dihybrid. It is a cross again between two pure homozygous parents, as Mendel always selected pure parents, in which here we are going to study the inheritance pattern of two pairs of contrasting characters. When we studied one pair of contrasting character, we called it as a monohybrid cross. When we are studying two pairs of contrasting characters, we are calling it as a dihybrid cross. Mendel followed the same 3 steps. First, he reared all the pure breed for 3 generations. Here he carried out a cross between yellow and round. These 2 characters he studied. One was the color of the seed and other was the shape of the seed. This was treated as a female plant and it was dominant. and the recessive one was green and wrinkled seed, green color and wrinkled shape of the seed, which was treated as male. He carried out hybridization, artificial cross, and it was carried out between these two parents with two pairs of contrasting characters. Of course, in F1 generation, what was produced? Dihybrids were produced, which had the phenotype was yellow and round, but the genotype was, it is a hybrid, heterozygote, capital Y, capital R, small r. We'll see this with the help of the chart also. It will be easy for you. From this, it was proved that yellow is dominant over green and round is dominant over wrinkled seeds. So heterozygous dihybrids were obtained. Then he carried out selfing of F1 generation. He obtained F2 generation. He found that yellow and round seeds produced were 315, yellow and wrinkled 108, green and round seeds were 101, green and wrinkled were 32. So total 556, which were in the ratio of 9:3:3:1. How? I will show you after this. Out of 4, 2 were parental combinations, yellow and round, green and wrinkled, and 2 were new combinations, yellow and wrinkled, green and round. He then carried out reciprocal cross and he's got the same type of results. Same combination but reciprocal cross now. At the end got the same phenotypic ratio 9:3:1 and the genotypic ratio was 1:2:2:4 1:2:1:2:1. You can also remember as 12:24 1:1:1. Okay, 12:24 1:1:1. Or you can remember as 1:2:1:2, 4 2:1:2:1. Let us see how this is obtained. Will this study this carefully? So he selected P1 generation. What was the phenotype? Yellow and round seed. What was the genotype? Capital Y, capital Y, capital R, capital R. He crossed it with another plant which was green and wrinkled phenotype. Genotype was small y, small y, small r, small r. Write with me so that you will understand. What are the gametes formed? Capital Y, capital R. Here the gametes formed are small y, small r. F1 generation, when they come together, we get capital Y, capital R, small r. Dihybrids are obtained genotypically. Capital Y, small y, hybrid. Capital R, small r, another hybrid, 2 hybrids. But the phenotype is yellow and round, so it is similar to the this parent, first parent. So here we are concluding that yellow color is dominant over green and round is dominant over wrinkled. Then it was allowed to undergo selfing. Now interesting thing took place. Please pay attention. When this was allowed to undergo selfing, these pairs, members in the allelic pair, they segregated from one another. Capital Y, capital R, small r, all separated from one another. Look here, capital Y, capital R, small r. They segregated with each other from each other. Segregation means separated from one another, and they assorted independently. They made partners with whatever partner they wanted to choose. For example, capital Y here came with capital R. See, capital Y, capital R, and if you can see, this is similar to the parent. Capital Y then came with the small r, small y with capital R, small y with small r. So this is very important. Members in the hybrid, F1 hybrid, they remain together. Members in the allelic pair, they remain together without mixing with each other. And while gamete formation, they are segregating from one another and they are independently assorting to form different partners. And now, what was the result of selfing? A result of selfing was, here we are treating these gametes as the male gametes and these gametes as the female gametes. Listen carefully. So capital Y, capital R, capital Y, capital R, yellow color, we are getting this yellow. Now I'll just write here yellow and round seeds. So yellow and round. I'll write the numbers. Okay, we'll count. We'll see how the phenotypic ratio is coming. Yellow and round 1. Second was capital Y, capital R, small r, yellow and round 2. Yellow and round 3, if you see capital Y, capital R, capital R. Yellow and round again 4, 5, 6, 7, 8, 9. You can draw a triangle like this. So these are yellow and round. How many yellow and round you are getting? 9. Correct? Now let us see the second case. That is yellow and wrinkled. I'll use another color. Yellow and wrinkled. Look at this picture. This is yellow and wrinkled. Why yellow and wrinkled? Because capital Y is giving yellow color. Small r is giving wrinkled shape. So yellow and wrinkled. This is also yellow and wrinkled. This is also yellow and wrinkled. So I'll draw a triangle with red color. How many yellow and wrinkled are we getting? We are getting 3. So yellow and wrinkled. How many are we getting? Yellow and wrinkled we are getting 3. Now let us have a look at the third case, that is green and round. Look here, small y, so it is green, capital R, so it is round. I'll use green color. So this is green and round, green and round, green and round. How many? 1, 2, 3. So our second phenotype is green and round. How many are they? They are 3. And the last is small y is representing green and small is representing wrinkled. Green and wrinkled, how many? So green and wrinkled. How many are we getting? 1. So what is the phenotypic ratio? 9:3:3:1. I hope you understood how he obtained this phenotypic ratio 9:3:3:1. I'll just give you a brief revision, students, if you don't understand again. Capital Y, capital Y, capital Y, capital R, capital R. What will be the color? Yellow and round. Capital Y, small r, small r. Small r is representing wrinkled. So the seed will be yellow and wrinkled, this case. So in this way, the phenotypic ratio obtained was 9:3:3:1. Genotypic ratio, I'll just erase all this so that you come to know about the genotypic ratio also. We'll just clear everything. Genotypic ratio means the genetic constitution. That also I'll try to explain the genotypic ratio. Look at the chart once again. Okay, how many plants are produced in F2 generation altogether? We have produced 16 plants. So let us see the 16 plants. Capital Y, capital Y, capital R, yellow and round. How many? Only 1. Capital Y, capital R, small r, yellow and round, but different type. Can you see this one? I'll draw a 1 this and this 2. So these are 2. Capital Y, small y, capital R. How many? 2. So I think this one I'll draw a star 1. And where is the other one? I think this, yeah. So this also is done. These are 4. So they are very simple. See, capital Y, small y, dihybrids. All the dihybrids are in a line. How many are they? 4. Capital Y, small r. Where can I see it? Yes, I think this is the case. No, not this. It is here. Yeah, this is the case. Okay. Then capital Y, small y, capital R. These are the cases. I'm ticking right. Okay. Then small y, and capital R. See, this is the case. I'm making a circle. Then small y. Look at these two. These are small y, capital R, small r. And finally this one. So from this also genotypic ratio, how much it is? 1:2:2:4:1:2:1:2:1. Simple way to remember, 12:24, 1:1:1. You can write it in the other way also. But this numbering and the genotype should not go wrong. So again I repeat, in dihybrid cross, the phenotypic ratio is 9:3:3:1 and the genotypic ratio is 1:2:2:4, 1:2:1:2:1. So I hope you understood 3 important things we concluded here. He selected pure plants. He carried out hybridization. F1 generation, he obtained dihybrids. The members in the allelic pair, they segregated from another, they assorted independently. And F2 generation, the phenotypic ratio obtained was 9:3:1, and genotypic ratio was 1:2:2:4, 1:1:1. He carried out the reciprocal. This first time he treated this as a female, this as a male. Second time he treated opposite, but still he obtained the same result. This is a dihybrid cross, can be asked in the exam for 3 marks, only a chart. You practice at home taking different examples of dihybrid ratios. Now we will discuss about the third law of independent assortment based on the dihybrid cross. When the 2 homozygote parents differing in 2 pairs of contrasting characters. What contrasting characters we took here? Yellow and round crossed with green and wrinkled. Across, the inheritance of one pair is independent of the another pair. Inheritance of this pair is independent of the inheritance of the other pair. When a dihybrid or a polyhybrid forms a gametes. Now what was our dihybrid? Capital Y, capital R, small r. When they are forming gametes, they independently assort, they segregate from one another first. So capital Y, capital R, small r. They are segregating from each other and then they are independently assorting, separating from one another so that different combinations are formed. Capital Y will form gamete with capital R. Capital Y may also form a gamete with small r. Small y may go and form a partner with capital R and small y may form a partner with small r. So what does this show? What was the original combination? Capital Y, capital R. This is similar to the parent. What was the second combination? Capital small y, small r. So this one is similar to the parent, but these two combinations are new combinations than the original one. This is nothing but law of independent assortment. However, this law is not universally accepted. It is not accepted in the case of linkages. What are linkages? What are linked genes? I'll teach you in one of the next lectures. So I hope you have understood third law of independent assortment, that members in a allelic pair, in a dihybrid ratio, in a dihybrid cross, they stay together. They do not mix with each other. They segregate from each other and then they independently assort irrespective of their parental combinations when they are forming gametes. Students, all the three laws are very, very, very important. Write them, read them and understand them again and again. The first law is not universally applicable. The third law of independent assortment is not universally applicable. Only the second law of purity of gametes is universally accepted. Recombinations occurs due to crossing over during meiosis. I will discuss about this when we will study about linkages. That time it will be clear to you. So this law is applicable only to the genes which are located on different chromosomes. Linked genes cannot follow this law always because there is a chance of separating. Hello students, we have studied so many important concepts up till now. How will you revise? Step by step. First we studied monohybrid cross. In monohybrid cross, we are one pair of contrasting characters we are studying. What was the phenotypic ratio? 3:1. Genotypic ratio 1:2:1. Then we studied dihybrid cross. In dihybrid cross, we are studying two pairs of contrasting characters and we are crossing two homozygous pair parents. What is the phenotypic ratio obtained? 9:3:3:1. And what is the genotypic ratio? 1:2:4, 1:1:1. Okay. You should also remember the three laws. First law was law of dominance. The characters that are expressing itself in F1 generation in heterozygote condition are dominant characters. The one which are suppressed are recessive characters. This law is not universally accepted. Second law was purity of gametes. Gametes stay together. The members of the allelic pair stay together without mixing with each other. It is law of purity of gametes, universally accepted. And the third law is law of independent assortment. The members of the dihybrid, the allelic members of the dihybrid, they segregate from one another and then they independently assort, forming new parental combinations that were different from the parents is possible. And this law is also not universally accepted. Please revise these laws, learn them again and again, practice some other examples for monohybrid and dihybrid cross. We'll continue our lesson next time. I hope you understood. Thank you.